to head Inferring frames, and I'm supervised by Sandrine Lagarig, who's a geneticist, and David Koser, a statistician. And today my talk is about gene network inference, and it's a common work with Magali Way and Chloé Friguet. So my thesis combines genetic and statistics, and the subject is the genetic analysis of a complex trait using transcriptomic data and the contribution of gene network modeling. So what is a complex trait? First of all, what do we call a complex trait? It's a phenotypic trait. It can be like the body weight. Why do we call that a complex trait? It's because it can be controlled by several genetical factors. So by transcriptomic data, the transcriptomic data are the gene expression data. So we have for one organism 1,000 gene expression profile. So the idea is here to find a genome region that controls the variability of the trait of interest. This kind of region are called QTL, for quantitative trait loci, and we want to find also the gene or this region. And what is interesting is then to model the gene network underlying the trait variability. So, and today I'm going to present you this part of my PhD. So basically, there is a true network that is most of the time completely unknown, and using this transcriptomic data and the statistical methods, we're going to try to infer the gene network. So that's the graphical representation. So by node we mean a gene, and an edge is a link between two genes. There exists two different types of representation: the undirected graphical model or the directed acyclic graphical model, or Bayesian network. But in this study, we're going to focus on the undirected graphical model, and there are two types of methods who are dealing with this model: the relevance network and the graphical Gaussian model. So the different steps are as follows. First of all, we choose a measure of the link between two genes. Then for all pair of genes, we're going to calculate this measure. So, and then connect the variable by an undirected edge if this measure is higher than a certain threshold, or if we can make a statistical test if the edge is significant. So here we can represent this matrix of measurements into a graph like this. Then the good question is, which kind of measure are we going to choose to find if two genes are linked? I think the most intuitive one is the classical correlation, the Pearson correlation, and this one was used in the so-called method relevance network introduced by Butte et al. In 2000. But with this kind of measurement, there is a confusion between direct and indirect links. So, for example, here, if we have a gene which controls both of these red genes, it can create a spurious correlation. And we want to represent in our network the direct link between genes. That's why there is another measurement, which is a partial correlation. So it means we're going to calculate the correlation between two genes, taking into account all the other environments, so all the other genes. And that is used in the graphical Gaussian model, and so it allows highlighting direct links only. So that's why in our study we choose to base inference on graphical Gaussian model. in this model, we suppose that Y, the dataset with n individuals and p genes, follow a multivariate normal distribution, and it can be shown that partial correlation is directly linked to the inverse of the variance covariance matrix. So, as follows. So, to calculate the partial correlation, we have to estimate and invert sigma, which is quite a challenging issue when the number of variables is really higher than the number of individuals. So, to address this task, a number of methods have been developed quite recently. And one of them was introduced by Schaeffer and Streamer in 2005, and they proposed a shrinkage estimator of the variance, the covariance estimator. So here, lambda is the tuning parameter, T is a target matrix that corresponds to a low-dimensional submodel, and S is the empirical covariance matrix. So the aim is to create an estimator that can be easily inverted. There is also an alternative route called the regularized regression. So we regressed all the genes against the remaining set of variables, and there is so we can calculate then the partial correlation simply like this with the coefficient regression. And as we suppose that most of high-dimensional problems are intrinsically sparse, most of them are using lasso techniques. And here you have three versions of those methods, so G-lasso, space, and Simon. So, all the methods I presented now are widely used in the biologist community, but an efficient estimation of partial correlation is still a questioning issue. And we propose here a new methodology based on factor analysis model. So this model is based on the splitting of the variance covariance matrix into two parts. A common one part containing the common information here and one part containing the specific information. And in terms of the data matrix, it's the same. Here we have the common information contained captured by factors that represent Z here. So into Q factors, with Q higher comparing, of course, to the number of genes, and a specific part contained in the residuals. And to estimate then the parameters of these models, we use an EM algorithm. So here you have the log likelihood, which is written like that. And in the E step, we estimate the expectation of the log likelihood using, estimating the conditional expectation given y of s, y, z, and s, t, z, and in the M step consists in maximizing the expectation given y of the log likelihood, and using this we can obtain an estimation of B and C. And what is really nice using this model for our partial correlation is that thanks to the Wedbury's identity, we can easily invert this decomposition of the variance covariance matrix like this. So it requires only the inversion of QQ matrix with Q really lower compared to the number of genes. And that's the nice trick of the methods. So then we propose here a new parameters, so phi and theta, in order to rewrite the inverse of the covariance matrix as a kind of factor analysis model where he did that plays the role of loadings. And so zero entry in the inverse of the covariance matrix corresponds to detecting data equal to zero. And using the theory of the maximum likelihood, we can then test the significance of data here using a student test. And also in the methods we take into account, we correct for multiple testing. So in order to evaluate the efficiency of our methods, we compare it with the most used methods, GNet and Sparse. And for fair comparisons, we use the default parameters for each method without additional tuning. So the simulation here of the inverse covariance matrix was sparse and with 5 blocks, as you can see here, so 5 modules of genes highly connected. We fixed the number of genes at 200 and considered different sample size, so 50, 100, 200 individuals. And for each case, we generated 100 microarray dataset. And we compare the 3 methods in terms of partial correlation estimation and in terms of edge detection. So first of all, the estimation of the partial correlation. So I reported here on this graph the estimated partial correlation versus the true partial correlation for our methods, the GNet methods and the Sparse methods, and for each sample size. So as you can see, first of all, is that as we increase the number of the sample size, the estimation becomes better, so that's really intuitive. We can also notice that for Sparse, this method tends to overestimate in each case the extreme partial correlation. And for low sample size, the GNet methods usually poor results. So that's quite surprising. And for each case, the methods based on the factor analysis gives a quite nice result. Now in terms of edge detection, I reported in this table the true positive, true negative, false positive, true negative specificity and sensibility. So what we call a true positive, it's a true edge detection, so a true non-zero entry in the matrix. And for each method, and here I distinguish the GNet with and without the multiple testing correction. So we can see that the specificity is really high except for the gene methods, and the sensibility is really, really poor for the GNet methods with the control of the FDR. So it's what we can see that the methods can well detect a few edge, but with a high specificity. And it's quite interesting to see that because there was a quite recent study that compared several methods, including GNet and Sparse, and they show the same results. So these methods are really good to detect a few edges, but with really high specificity. But in this case, again, we can see that the best compromise between specificity and sensibility is given by our method. So finally, we apply our methods in a study that aim at better understanding the genetical factors controlling the abdominal fatness variability in Michigan. Yeah, why? So that's a really important goal in the genetical lab at Agro Campus, so in my university. because it's a trait with a great, an economic trait, really important, because the abdominal fatness is not variable in chicken carcass, so we want to decrease the amount of it. And also, in a fundamental point of view, it's to better understand the regulation mechanism in the lipid metabolism process. So in a previous study, we highlight a genome region along the chromosome 5 on the chicken, so corresponding to hundred of genes and including the causal mutation, which is unknown. And we show that 59 genes were controlled by this region. So it was really interesting for us to model then the gene network of those genes and trying to find maybe some biological process underlying the adiposity variability and also to better characterize this region. So using our methods, we infer the gene network, and it's really interesting. We extracted two gene modules, and thanks to the annotation available, we managed to interpret functionally these two modules. And one of them was characterized by genes involved in the lipid metabolism, and particularly we deleted these genes, the HTR7, which encodes for the last enzyme in the synthesis of the cholesterol. So, and that gives really new hypotheses of the function that are involved in the fatness variability, and also about the causal mutation in the region. So to conclude, we show the efficiency of the factor analysis model for inferring gene network in the graphical Gaussian model context. And now we're trying to extend the comparison on other simulated design. So in changing other parameters like the density of the network and also the number of genes. We show that thanks to the gene network modeling, we found new functional hypotheses about the causal mutation and the biological process involved in the fatness variability. And we found now, in the meantime, other regions in the genome controlling fatness variability. So now it would be interesting to model the network for all those regions. So then here are some references of the methods I mentioned, and thank you for your attention.
Perfect. We have plenty of time. Questions, comments?
When you look at these networks, they look like small words somehow. So you have hubs, so a few genes that are very highly connected to a lot of other genes, right? So I'm curious if you, the method that you tried actually reveals this kind of structure, or maybe not in this particular application, or and also how this could affect the complexity for the inference, the graphical model, because if you have something that connects very highly everything, maybe the E-step of the EM algorithm.
Okay, so just you are surprised because there are not a lot of genes in the network?
No, I'm asking just whether you observed that or not.
So here in the example I showed you, so there is not a lot of genes that are controlled by the regions. So maybe that's why the network is not really big. But generally, it has been shown that the gene network are sparse. So there are not a lot of connection in all the genes involved.
No, that's not what I mean. Of course it's sparse, but still you have a few genes whose degree is very high.
What?
The degree in the network
The
of these
degree.
genes is
Yeah,
very high.
There are hub genes that we call
Hubs.
hub genes. Yes, and those genes are particularly interesting for us because that can be, the genes that are really important for the variability. Yes. So, yeah.
So you observed it.
We observe that. Yes.
So you predict
But then
this
I think.
hubs,
Okay, so there are really different parts in this gene network inference, and there is a big part after the graphical representation is the analysis of the graph. So here I didn't speak about that, but of course then you can analyze the network in terms of modules and hub networks and find these genes.
So you could come back to your picture with the graph. At least maybe that was a simplified graph.
So, yeah.
I think it gives a good idea of the general situation.
And
Yes, like that.
And so here we don't have all the genes because so we tested the significance of the edge, and I choose a threshold. So here it's 5%, and of course if you change the threshold you can have more genes, but yeah.
So how can you explain that this DHCR7 is so influent? It's very isolated.
So after that, you can, use the literature to try to understand biologically the network and the link between the genes. And there are also some tools that can be used complementary to this network. There is like Ingenuity, it's a software which model the gene network but based only on literature. So it's really interesting to compare the gene network we get using the gene expression and this kind of network. And this comparison can help us to understand what we find here.
Can you comment on the fact that genes are in general thousands?
Yes.
So, and now in the example you have just 59 genes. What do you do when you have 1,000 of genes and you have to make this kind of analysis? And also the computational complexity because you use EM algorithm.
That's a really good question, and for the moment, it's a challenge to do that for all the graphical Gaussian model methods. So that's why. So then it depends also what you want to do with the network. So what is your goal at the beginning? So for us, it's really to understand this kind of region that control a phenotypic trait, and those regions are controlling hundreds of genes, but it's really rare to have a genome region that controls thousands of genes. If you have that, it means that it's not so a complex trait that you have, but it's a trait controlled just by one factors, one genetical factors. So, but if you have another goal, like to understand all the mechanism for all your genes, actually these methods we have to improve the methods, but not only ours, but all the methods have this limitation.
And how do you select the genes?
So in our case, we select the genes which were controlled by the region. So, and that's another question, how we found these genes. So it's using linkage analysis. So that means we're trying to find eQTL, so it's no more QTL, but region that control the expression. So we found eQTL, and then to find whose genes are controlled by the particular region, we find the QTL, the EQTL who are colocalizing with the QTL. And that gives us the hypothesis that these genes are all controlled by the same mutation. So there was a big study before to find all these genes before the gene network modeling. Yeah.
More questions?
Is gene a discovery, or was it only
You mean in general, or like they are involved in this?
Is this gene generally involved in fatness?
Okay. We have some biological information, some annotations. So yes, for DHCR7, we know that it's involved in the cholesterol metabolism. But then in fatness variability in chickens, yeah, for the moment it's new. Yeah.
Okay. Then we have to thank you